All right, we can get started. So welcome to the mid-morning session. This is a session about shape statistics, but I think we can view it more generally as a session involving tools from geometry and statistics together, not necessarily just any technical definition of what shape is. We have some very good speakers. In fact, the first three speakers of the session are the people I have looked up to growing up working in the field. So I think we are very lucky to have.
Speaking too much.
Let me finish praising you first. So we are very lucky to have this high quality of speakers, all three of them here. So the first one is Professor Laurent Younes from Johns Hopkins University. So go ahead, Laurent.
Thank you. All right. Okay, so, well, thank you to the organizer for the invitation. I'm very happy to give this talk here. How much time do I have?
Well, now you have 14 minutes.
So I probably have to start wrapping up the. Okay, so I submitted my talk yesterday, and I realized that I forgot to include the introduction slide, but which is better probably because I would have lost time on the introduction. So I'm starting directly with the program, which is about comparing images. So we want to discuss, well, I still need to introduce a little bit what we're going to do. So we want to discuss image registration under a slightly more general framework that what is standard image registration, and then see how this can be adapted to comparing images of different modalities. So when you design image registration method, you always have to include somewhere some term that decides whether your images are similar or not. And that appears in every image registration method, whether it's as it should be LDDMM or something else, like deep learning. The term that we find very often are norms that compares the values of image as homologous points, and the basic example is an L2 norm in that setting. Then once you have this, the standard approach is to have some kind of regularization term that I won't talk about in this talk, but if it's LDDMM, it's a Riemannian distance on a diffeomorphism group, and this term that compares images once you apply the registration to the first one. So there's a few issues. I mean, a limitation with this norm. First, they are designed to compare vector valued function. If the function has its values in a feature space, or if it's a binary function, I know that this is applied very often to compare binary function. Binary function should not be compared using L2 norms. That's usually the 1 and the 0 have no meaning in terms of value, and the difference are misleading. Even gray value function for brains, for example, are not. I mean, there could be regions with similar gray values, but that have very different functions or interpretations. And so being able to use arbitrary values in arbitrary set is an important thing when you compare images. And of course, the LP norms don't make sense here, that there are, of course, different options that already have been used in the literature. But here I want to discuss the presentation of images as measures. So if you take an image that from some space omega to some space F, omega is a subset of RD, then you can easily associate to it a measure on the product space. Yes. The product space RD times F by this formula, which is just measuring for the fraction of the values of the image that are in the set capital F for the points that belong to capital A. And this things provides a representation of images that are defined on arbitrary subsets of RD, taking value in some feature space. So when you do that, you're fairly general, and so the only thing is that now you have to define how to compare such objects, and there's a bunch of places in which distances between measures are defined. The one that we like is a dual RKHS norm, which you can also find in the literature as maximal mean discrepancy. And it has a closed form formula once you have a reproducing kernel. It's a bit, I mean, I don't want to comment that formula. It's a dominant table with respect to the measure, and there is a typo because that should be nu and not mu here. That should be the same as here. Okay, so this is what we use, except that, well, if I go back to the formula we started with, there is somewhere the definition of how diffeomorphic transformation transforms an image that appear in the objective function, and so we want to extend that to measures, and so you can work on the definition of measures that are associated with images directly and make a simple change of variable in space, and you end up with the formula that is at the bottom here. And so you get, why did it move to the right? Well, you get a definition of the action of diffeomorphic transformation on measures that take this form. So you have the Jacobian determinant of the deformation that appears here as an extra weight in terms of the measure, and you have in space the action of universe on the considered set. All right, and then, well, once you have that, you define a variation of the previous problem where you compare measures instead of images using their L2 norms, and then you deal with it regarding the implementation. There would be a lot to say about how to implement that, but it's doable. You can use particles, you can use meshes to implement this kind of method. And I'm just going to show you a few results on third example mostly. So say that this is your template, this is your target. So you have a template which is a relatively clean object, and your target that is, well, some variation of it with some noise, so this is the transformation of the template that you find, which acts by reducing the size of the inner circle and the size increasing the size of the outer circle. You can do it in 3D. It's just to show that the method scales to an extent. You can do it on spatial transcriptomic images. This is actually what that was the target application of designing these methods. So a few words on spatial transcriptomics and MERFISH. It's data sets that provide the gene expression of cells together with their position in space. And so they provide very rich information on the tissue the tissue that is analyzed. And these are fantastic images. I think that was Nature Method of the Year in the year 2020 or 2019. I'm still wondering how to use it, and because you have to show that you can do more than in single cell expression, for example, and so that the spatial organization is really useful. But whether you know how to really exploit it or not, you have to also find how to map two different objects. So you can do it within a single brain. And so what we're looking here is a section of a brain compared to another section, so at the beginning and the end of the sequence. And that's for one gene. I think we were using in the registration something like 30 genes, selection of the set of all genes that are available to MERFISH. And so you can do that with different genes. I'm not going to run all the videos. By the way, Anuj, I have less than 4 minutes. If you...
I'll remind you.
You can do that with different brains. And so you have stronger registration. And as often you want to analyze the results, and one of the way you analyze the result is by computing Jacobian determinants of the transformation, and so you can get that. So I'm reaching now the part that was announced in the title, which is how do you do cross-modality mapping. And the good news is that when you're using measures, there is a very simple way to pass from one measure to the other, which is using transition probability. So the measure that we're using are measures on space times feature space. So space is space, and features here, when you do cross modality, they are different from one image to another. So you just need to find some good transition probability going from one feature space to another. And this is what we try to estimate here. And so that makes the problem only slightly more complicated, in which you don't even have to comment the formula. The only thing that you want to see here is that in that formula, you have an extra term here that I'm calling pi. So this rho and zeta define the space feature measure. I didn't really mention that, but you're given a measure of a feature space. You can decompose it under, well, it has to be a Polish space. I mean, you can decompose it as a product like that, and you just add a transition probability that goes from one feature space to another, and you get the a problem that minimizes now two objects. So we're fairly straightforward in the way we minimize this. We use alternate minimization, and here is an example here with some toy data in which you have an atlas, which is the part that is shown in green, with a green square around it, so that's the atlas. Oh. There it is. And you have a target that you don't observe. You observe that. So green is what is observed, but these are the targets that you observe, and you have for the template some kind of true latent classes that transition that you want to estimate, and that's what the transition probability provide, and they map correctly to the target. So this was a test example. We ran that on some atlases. So here, I won't comment that because I have 55, 54, 53. Okay, I won't comment that, but this is a situation in which you have an atlas, so no information at all, and you have a Murphy's image, and you want to map the labels of the atlas to the Murphy's image by estimating this kind of transition probability. So I think I'm done. These are the references. I have 30 seconds to answer the questions.
That's my job.
No, not these questions, but the three questions that were raised to us, and I forgot what they are, but there's something about what I'm going to do in the next 5 years. I have no clue, and I could make up something like everyone else, and I'm pretty sure that it's not what is going to happen. So I'm not answering that one. If I knew, of course, I wouldn't tell you. How can we make people in imaging love us? I mean, love us statistician, including myself as a statistician. It's maybe a fraud, but let me try to do that. It's complicated. I mean, especially those who are not statisticians usually have been trained in statistics, and the way you learn statistics in most colleges around is dreadful, and so it makes people start with a very strong bias against any person who would like statistics and not to make it their job. This being said, yes, what was said before, offering easy to use data and algorithms, making them available in a way that can be used by user is really important. It's also important not only so we all publish our data, and when you have raw data, we publish the whole raw data. Most of the time, the data in the papers are processed, and it's important to publish the processed data in addition to the raw data, so that the person that is going to try to use your data doesn't always have to start from scratch to do it. I know Hong2 is doing it by himself, but normal people really don't have the time. Okay, I don't remember what the third question is, but I'm late already, so I can stop.
All right, thanks very much, Laurent. We won't have time for any more questions because he used up his questions. All right, the next speaker, let's get him online, is Lakshmi Mahadevan from Harvard University. Now, Maha is a guy I can introduce for 15 minutes, and then we'll have no time left to talk. Good to see you, Maha.
Nice to see you, Anuj.
All right, so I'll let him talk about his things. He does lots and lots of things. I highly encourage everybody to visit his website and the videos and
That's
all.
enough. That's enough.
All the media publicity that is covered on his research. So go ahead, Maha.
Okay. Thank you, Anuj. Can people hear me?
Yeah.
Yes.
And you can see my screen?
Yeah.
Okay.
It's too small.
Can you see it now? Better?
Better. Yeah.
Okay. Thank you. Thank you, Anuj. Thanks to the organizers. I'm sorry I'm not able to be there. I have a very strong sense of imposter syndrome. I'm not quite sure what I'm doing at this conference, even if it's virtual. So I will tell you what I'm thinking about, and I'm hoping that this will be mostly associated with questions that I imagine all of you can help with. So rather than think about it, the problem, what is the problem of understanding brain shape or brain connectivity on a fine-grained scale, I want to ask questions on a very coarse-grained scale. And I want to pose this from a generative perspective as well to see what we can also say about the processes which lead to shape complexity. And I'll focus on the brain. We've looked at other problems of this kind in morphogenesis in general. The morphometry will be very small, will be a very small part of the talk. So I want to just remind people of the fact that one of the great icons of complexity in biology is the shape of the brain itself, but all brains are not equally complex. This is a very nice image taken from a review about 15 years ago or so This is the human brain, the mouse brain. Most people who work in a laboratory setting, of course, have focused on the mouse for obvious reasons. But the interesting question is associated with the convolutions of the human brain. And the human brain folds in a non-trivial and non-linear way. So this is gyrification index measured, for example, as a ratio of the actual area relative to the area of the convex hull as a function of time here in weeks. So the brain starts to fold at about between week 26 and, week 20 and 24 in the human, and then continues to fold past birth and then saturates at about age 2 or so, and then more or less flattens out. And this is, as I showed you, as you can see on the left, it's not true for all brains. Functional consequences we all know, we all accept, and this is backed up by fantastic neurological, neuroanatomical data associated with the fact that the folded cortex, which is a fairly recent evolutionary innovation, maximizes cortical connectivity while simultaneously minimizing wiring length by essentially crumpling the surface of the brain. The two hypotheses which are thought to have driven this are sort of diametrically opposite, and one of them now we believe is the most dominant one. One is that the surface is growing relative to the bulk, so the gray matter is growing relative to the white matter. And the second one is that instead of the gray matter growing relative to the white matter, the gray matter is pulled inwards by the white matter. So if you essentially have axonal tension in the growing cortex, that could essentially pull the surface and cause it to fold. Over the last 20 years or so, we now know that the second is almost certainly not correct, that it's primarily associated with increased surface expansion of the cortex, which can happen for 3 different reasons. One is you have migration from the white matter into the gray matter, so you have more surface. A second is that the gray matter itself is proliferating. And then the third is that you can have the cells remain the same and the size change. And in fact, the second and the third are not so important. It's the first that's important, namely a cell migration from subventricular zones. So that then changes the problem from a biological one, at least in a coarse-grained way, to a physical one, asking what kinds of shapes will you get when you have constrained growth of the cortex, the constraint associated with attaching the cortex to the white matter, but then the surface is expanding because you have migration from the core into the cortex. And this should not surprise you at all, can give rise to these folding patterns, such as what you would observe. You put your fingers in water or go to swim in the ocean for a long time, come out and you see your fingers wrinkle. And so this wrinkling is associated with the fact that water has entered the upper layers of your epidermis and then it increases the surface area. How can you explain that? So this is kind of old work, but I want to set the stage over here. We can do an experiment where we can take a gel, we can put it in a solvent, and if it absorbs liquid from the solvent, in this case, the gel is polydimethyl siloxane, the solvent is hexane, and as it absorbs solvent from the surrounding, the surface starts to swell, and the physics we can understand fairly easily. The diffusive process is relatively slow compared to the elastic equilibration, which is fast. So we can ignore dynamics associated with elasticity. And if we think about quasi-statically swelling it, we're trying to minimize the elastic energy associated with differential growth of one part of a solid compared to the other. And that differential growth I can decompose into a part which is elastic and a part associated with growth using this multiplicative decomposition of the map which takes you from the reference state to the current state. So a deformation, effectively very similar to what Laurent Younes talked about in the previous talk, except now adding a contribution which is or thinking about it as a contribution from an energetic point of view. And minimize an energy, which I will not get into. It's a fairly simple energy associated with describing elastomers. I'll show you a movie of what happens. So this is a computation, and if the surface grows relative to the bulk, you start seeing these folding patterns, which are not dissimilar, at least, to what you see in the brain. And you take a cross-section, you see what I think everybody over here has seen at some point or the other in MRI-based images, and you can do the same thing experimentally by taking one of these gels and putting it in a solvent, and if you allow the solvent to gradually get imbibed into the solid, then you can see over time that you start forming these sulci and gyri. Sulci are these steep creases, and gyri would separate them. Okay. We want to now take that and see if we can make sense of it in the context of understanding the human brain. These are images, MRI images, actual images from museums of fetal brains, and you can see that the folding, as I said, starts at about between week 20 and 25, and then proceeds and continues after birth. So using MRI data, you can essentially create a physical model of this, just like the gel that I showed you. And I just wanted to show you a video of what happens in 10 minutes in a way which allows us to try and mimic what happens in 20 weeks. So this is a gel, it's not a brain. All that's done is it's shaped in the form of a human fetal brain to scale, dipped in solvent. And if you dip it in solvent and you wait for about 15 minutes, you start seeing the formation of the folds. And I'll show you an image in just a minute of the static outcome. And here it is. So you're able to capture it. So how can you make sense of it? First, from a physical perspective, I already told you, you can essentially construct a developmental mechanical model where the surface is growing relative to bulk. There is a parameter over here which tells you how much surface expansion is associated with the change in the metric of the surface compared to what's happening in the interior. And there is a characteristic scale over which that's essentially localized. It doesn't happen everywhere. So it happens primarily at the surface and decays into the interior. And this is a functional form. And of course, the idea would be to see how that varies as a function of space and time. And in this case, there is no variation in space, and the variation in time we also ignored, essentially just thinking about it as a quasi-static picture. And this is a computation that happens that you can do, solving the minimization problem that I told you about. And from that, you can calculate computationally the gyrification index. So in dots are the actual data. In small dots, the simulations are associated with the blue squares, and the red is the gel brain. The gel brain, of course, can't go forever because once the entire gel swells, when the solvent is imbibed into it completely, the brain just becomes one uniform, large version of the small brain. So there's essentially a similarity transform connecting the original to the final shape. So you get nothing at all at the end. It's completely smooth, and so you have to stop over here. You can do a little bit of morphometrics by mapping the surface of the brain onto the surface of a sphere, and then use a Laplace-Beltrami, the eigenfunctions of the Laplace-Beltrami operator, to essentially map the folds from the surface of the brain to the surface of a sphere, and you're able to do a reasonable semi-quantitative comparison. I won't get into the quantitative aspects. I'm happy to answer questions later on because I wanted to give you a very coarse view. Okay, you can then, how much time do I have, Anuj?
You have 8 minutes.
Okay. So then you can ask, can I construct this morphospace? I've studied it in a simple gel. We've studied it in the context of an attempt to try and understand what happens perhaps in the human brain. Can we now zoom out and think about it as a function of what the brain folds look like across species? And in the simplest setting, when I don't worry about spatial variations in the metric, which tells you what the expansion coefficient of the cortex relative to the bulk, so I don't worry about the spatial complexity at all. I average over the entire brain. The vertical axis tells you about tangential expansion. The horizontal axis tells you about the thickness of the cortex relative to the size of the brain. So if the cortex is thick relative to the size of the brain, it's unlikely to fold because it's very thick. Therefore, energetically, it's extremely difficult to deform. And if the cortical expansion is relatively small, then it's unlikely to fold. So we know that from the biological literature. Rat brains, at least the rat cerebrum is not folded, the rat cerebellum is folded. And as you go along the vertical axis and the horizontal axis, you end up with more tangential expansion and a thinner cortex. The horizontal axis is the size of the brain relative to the thickness of the cortex. So if the cortex is thin, it's much more likely to be flexible and therefore much more likely to fold. And so you do see this natural characterization. The solid curve is data from neuroanatomy. The bottom shapes are computations, and of course the top ones are a qualitative sense of what you see as you go from one organism to the other. A question to which I don't have an answer. I want to pose this mostly as a question, and the coloration is mostly as a hopeful guide rather than anything else. again, this community knows much more than I do about functional specificity. Very important question in the context of brain function is understanding localization. Could it be that the folds actually, as they come to be, through this iterative mechanical instability, lead to specialization? Rather than say that specialization is early or specialization is late, could specialization and folding develop simultaneously? I don't have an answer. That's an experimental question, primarily because we have to study how function develops even as the brain folds. An extremely difficult question experimentally to address. That's a thought. It's only a question at this point. We can explain some of the pathologies that people have observed in the context of seeing when the folds become smaller. So polymicrogyria, I'll come back to this in a genetic context in just a minute. Large, fine folds arise when the cortex is thin, as you would expect, pachygyria. So if you have a thick cortex, then the folds will be much more widely spaced. That comes out of this very simple physical picture. And lissencephaly, the absence of folds, like what you see, for example, in children whose mothers have been infected by Zika virus, for example, very small or no folds, and that's associated with the brain itself being very small. That means that the cortex is effectively very thick. So that's qualitative. Can we understand this genetically? So a very good model system, in fact, perhaps the only model system that exists to understand cortical morphogenesis is the ferret. As I said, as you know, the mouse brain does not fold. At least the cortex doesn't, cerebrum doesn't fold. So you have to look for another organism, and the organism of choice is the ferret. Here is, and why? Because the ferret brain, the ferret pup, is born with a relatively smooth cortex, and over a period of about 30 weeks, it progresses from an unfolded to a fully folded cortex. So you can essentially examine this using MRI data and more invasive processes. And so you can redo these experiments that I just showed you. This is very recent work. Hopefully will be in the archive in a few weeks. Start at different stages, and then you can watch going from top to bottom how the folds form. These are different views. You can also do computations. So exactly the same way as I told you about, which then begs the question, I hope let me just remove that. So can you do stage simulations at different stages? Because you can essentially arrest that folding pattern, take MRI, not arrest it, but take MRIs at different stages and then use those different stages as initial conditions to go from, let's say, P4 to P8, P8 to P16, P16 to P30, and so on. And you can capture that. And again, you can use, again, from your perspective, from this community's perspective, perhaps very naive and extremely simplistic ways of characterizing the morphometry by, again, mapping the surface of the ferret folded brain onto a unit sphere, and then have some landmarks associated with specific folds, for example, the prosylvian fissure, the central cortex, and so on. And again, I'm not going to get into the details of this. So you can start capturing some of it, and you do see, again, qualitative similarities. The question of quantitative similarities is something that we aren't comfortable yet because we don't have good enough sample sizes. Okay, I'm going to finish by saying something which I think is particularly exciting, at least for me, which is associated with genetic manipulations. So collaborators of ours in the medical school here from the lab of Chris Walls have shown that there are specific genetic mutants, in this case SCN3A, which you can localize using electroporation. And then you localize, when you localize them, you start to see formation of polymicrogyria, which shouldn't surprise you, you can capture. You look in the literature, there are other defects, again, in the ferret associated with cortical migrations. That means less cells, neurons migrating from the inside to the surface. A consequence of that is you get less expansion, and you also have microcephaly. Okay, I will finish by asking a question which we are just beginning to start thinking about. I've shown it to you in the context. I've shown you something associated with how cortical folds form in the human brain. Now starting to think about it in the ferret, because it's a very good model system to connect what's happening genetically to what's happening on the macroscopic scale. Can we now zoom out and think about different species? And the species that we've chosen are associated with experimental accessibility. The ferret, which I told you about, the human, about which we know a lot, and the macaque, which has been a subject of some study. I will not go over the last slide, but I'm going to finish by telling you where we are. So, I think there was a question associated with what are the questions that excite me from a slightly different perspective than I think the community that perhaps I am addressing. First, we need more cell migration and proliferation data to make this idea of mechanical instabilities, which seems to be developing some traction and is consistent, not just qualitatively, but starting to become quantitatively correct as well. Second, very interestingly, there are mutants which have pathophysiological outputs, which either change the size of the brain, the thickness of the cortex, or the expansion. And those are fundamentally the only parameters in the physical model that I presented. I've given you some examples over here. Again, can we make this quantitative? And perhaps the most interesting and enticing question of all, which is the one that I raised with that image, colored image, how can we start thinking about the simultaneous process of folding and functional specificity in both the normal and in the pathological brain, either in humans or in animals. Okay, all this was work done with students and postdocs, Thomas Tallinn and Jun Chang, who started it, Gabriel Sejourne, who's an undergraduate, who helped create some of these gel models, and most recently, Gary Choi and Stefanian. Thank you so much.
Thanks, Maha. Since we are running short of time, I think we'll postpone the questions. I do have a lot of questions, so I think we'll talk separately.
Okay, thank you.
Thank you, Maha. So the next speaker is, go ahead. You think we have time for a question?
It depends on.
Yeah, I think we are running a little late, so. So the next speaker is Xavier Pennec from INRIA, Sophia Antipolis, and University Côte d'Azur, and he's going to talk about a lot of different things. Go ahead, Xavier.
Okay, can you?
Oh,
Yeah.
Can you hear me? Okay.
Yeah.
Thanks. So it's very nice to be there, and it's very difficult to talk after the previous talk because I'm going to talk about mathematics. And I first want to state that this is a joint work with my PhD student, Jan Tanvelas, so most of the recent results are due to him. So the first thing I want to stress is the importance of geometry in data analysis. And one of the example is the example of shapes that we have seen with the previous talk and the talk of Laurent. So if you take, for instance, the surface of the brain and you want to do statistics on the shape of it, maybe you want it to be invariant with respect to the position and orientation in space, or with respect to the parametrization of the surface. So it means that in that case, you're going to consider the quotient space or the space of class of equivalence of shapes, and do what we call quotient geometry. And even if you start with something which is Euclidean flat space at the beginning, by doing this quotient, you're going to get into something which is curved. And here the problem is how to do statistics on these curved spaces. So for this talk, what I want to look at is covariance and correlation matrices. And here we want also to enforce some types of invariance. And most of the case, it will give also curvature to the space. And the question maybe that we should ask ourselves when we start doing statistics on this data is, to which space does the feature belong? What are the natural invariants? And then maybe what are the metrics that respect that? And then we can try to use a simple statistical algorithm or machine learning algorithm like k-means and things like that. So if you want to learn about that, there's a book where we put some of these notions and examples in medical image analysis. So for this talk, I want to focus on symmetric positive definite matrices, SPD matrices. So I personally worked on them for diffusion tensor imaging. So that's we did quite a lot of work for estimating, regularizing these images, but these are also features or data spaces that are used for brain connectomes and brain-computer interfaces. Here it was shown that this can have
quite an effect, or changing the metric can have an effect. So first, what's the problem with the classical Euclidean metric? So here is an example of what is the space of two-dimensional SPD matrices. So what you can see is it's first a subset of symmetric matrices, but the determinant, the positive determinant constraint put a cone structure. So what's happening is, well, it's a nice space. If you take two SPD matrices, you can interpolate in between. You will stay in the space because it's convex. But as soon as you're trying to, well, optimize something, you're going to shoot. You have a lot of chances to hit the boundary and get out of the space. So, so how to change that? Well, one of the ideas is if we don't want to reach the boundary of the space here, maybe we should penalize things that go close to the space. So if we think about a curve, maybe we should say it's more and more difficult to approach the boundaries as we get closer and closer to the boundary. And this idea of penalizing the acceleration or putting a cost on the acceleration is exactly what is done in Riemannian geometry. And instead of straight line, we're going to change that for a path that minimize the energy or the length of curves. And so these are the mathematics. You don't need to understand everything. I'm just saying that we mathematicians are there to solve this for you. But if we specify a scalar product, then we can compute the geodesics with this second order differential equation. The only thing that we need to understand from there is that there is a local solution from any point with a tangent vector. So it's called the geodesic shooting. That's what is called the exponential map. And the inverse, finding the vector that allows you to shoot from X to Y, is what we call the log map. If we can implement these two operations, then we can do, we can generalize many algorithms. And this is a list of the type of things that have been done so far. So there's obviously Fréchet mean, Riemannian optimization, central limit theorem, extending PCA to manifolds, and maybe things like domain adaptation. If you want to compare what's happening with different part of your space, then you can use power transport to do that. Okay, so let's go back to SPD matrices. So if we want to look at what is a natural constraint on these matrices or natural invariance, we can look at what is the effect of the action of an affine transformation. So if you change the basis in which you represent your matrix, then the action will be basically a linear or matrix acting by conjugation like that. And we may say, okay, my distances, I want them to be invariant if I do, or if I compute the distance before or after this change of basis. Okay, so this, I want to have an invariant action. So if we do that, then we can show that basically we can use a transformation sigma minus 1/2. So it means that we can characterize the scalar product by its value at the identity. And moreover, this scalar product should be invariant by rotations. And all these scalar products are of this form here. And we can determine that, well, in closed form, what is the expression of the exponential and log map. So what's nice here is we can implement things. I mean, the formulas by themselves are not important. The only thing is we can put that in the machine and it's going to compute. And if we do that, if we look at what is the change, so if we look at the interpolation from this matrix here to this one, this is here what we get with the Euclidean metric here on this side with the Riemannian geodesics, and it has different properties. For instance, the trace is monotone with the Euclidean metric, while the determinant is monotone with the affine invariant metric. So these are the type of things that may be important to investigate on your data. Does it make sense to have this type of monotonic interpolation? And it had a big effect on, for instance, the processing of DTI images. But it was also shown that it could make a big difference, for instance, in classification of brain-computer or BCI signals, for instance, in the paper of Parachon. Okay, so there are other metrics like the log Euclidean. So it's the log Euclidean can be seen as somehow a push forward. So we take a Euclidean metric on the space of symmetric matrices, and we take the exponential, and then we get something on the space of SPD matrices. So it's a different metric. And it can be shown that it's actually a metric that approximates well the affine invariant metric in each tangent space, depending on the Euclidean metric that you choose. And more generally, there's families of metrics that can be designed. So they can be related with alpha beta divergences in information geometry. But we can see that there are relationships between them, and we can try to, or we can think of optimizing which metric would be the best for your data. Okay, I'm going to skip that. There's even larger number of metrics that were proposed. The only message is there is a way with invariance to try to classify them. And I want to go to the correlation matrices, which is maybe the new part of what we're doing. So usually what is done is we've got in fMRI analysis, for instance, we get the correlation between two regions, but the variance is something that is a variable of non-interest. So basically we want to normalize by the diagonal. So we normalize by the diagonal on both sides here. And this is something that is done in many different areas of data science. And if we look at what is the space in which we end up, so this is for the 3x3 matrices, the space of correlation matrices. So you see that this is something which is compact. So I like it because it has the shape of a candy in France, which is called a bell angle. But it's even worse than the cone that we had before, because now if you're anywhere and you're shooting in a direction, you're sure to hit the boundary. So one of the questions is, can we change that? There is an answer, which is to say, yeah, if we don't start with the Euclidean metric, but we consider this quotient space by the action of diagonal, if we start by the affine invariant metric, then we can design and we can show that there is a well-posed metric in the quotient space, which is called the quotient affine metric. And it's a geodesically complete space. There's no boundary. And it has some other properties. And after a long work, Jan was able to establish the formula for the exponential map. It's not, once again, it's just the fact that we can implement it, which is important. And more recently was even able to show that there are unique geodesics. So the thing is, the log itself has to be computed by optimization. And that might be a problem because here there's positive and negative curvature. So we know that numerically it may be difficult to do, and it's time consuming. But if we look at one of the examples here, so we took two SPD matrices with correlation coefficients that are here. So these are in 2D. The interpolation we get with this metric for the correlation is monotonous, while you see that the affine invariant is something like that, and the log Euclidean is not even going to the same value in the end. So what's interesting is there is a different interpolation in between, but the extrapolation might be very different. So if you're thinking of extrapolating from your data, maybe changing the metric will change a lot of things. Okay, so what I said is the log has to be obtained by computations or by optimization. So what we try to do is to do the log Euclidean trick, try to find some kind of Euclidean approximations of that metric. We were able to propose 3 new metrics, so I don't want to enter into details. The only thing is they have different properties. They are currently being implemented in the Python package geomstat, where we are trying to have a place for reproducible computations with geometry. And the idea is to have, we have a first module where we put everything about the manifolds, the way to construct manifolds, Riemannian manifolds, and metrics on them, and quotient metrics and things like that are implemented in there. And if we can put the exponential and log map, so if we don't have the formula, then we can have optimization. So this can be computed, even the log map can be computed by automatic differentiation, but that's going to be very costly. If you've got the closed form, then it's going to be much faster. But anyway, whatever the manifold we have, if we implement the basic operation here, Then we can use the second module, which is here, which is all the algorithm that we can design, and they're going to work automatically on any of the metric that you choose. So maybe one of the message here is, well, we're actually this guy here, if I can, yeah, Olivier Bisson is the one who is currently implementing all that. And in a few months, the metrics that I just showed you, they should be available in GeoMstat, and you can try to see what it gives. So maybe to finish a few challenges from the mathematical point of view. So first, the first message is if we're computing on a space of correlation matrices, maybe it's a good idea to try to look how to respect the structure of that space. So if we have correlation matrices, we want to interpolate or extrapolate, then this should remain a correlation matrix. Somehow, if we want to keep the interpretation. So that's the mathematical setting. Second thing is with different families of metric, maybe we can think about optimizing the metric. So there's a huge work in metric learning. Here, it's a very constrained problem. It's if we have some kind of invariance, which one is the best for our data? And maybe one of the idea that is behind is we could use the geometry or the invariance property to choose what is the main class of metrics that is interesting for our data. Then we could try to design a log Euclidean approximation to make it work efficiently. And now if we are in the flat space and our data are not too badly represented, then we can use neural networks to try to fine-tune the smaller nonlinearities. But maybe using geometry as the first tool to find what are the large-scale nonlinearities is a good idea to start with. And maybe the third point that I wanted to make is, well, there's packages to share these things. So it should be easy to test and to compare metrics, and it should be hopefully reproducible, because if we use all the same algorithms and if they are well designed, then they should be reproducible science for this type of analysis. And with this, I want to thank the people of my group for the last few years.
Actually, we have time for a question. Xavier left some seconds. Yeah. All right. Any quick questions? Just have time for one question. We'll let Mo ask this one.
So if low computation is
hard, can you just do the tail expansion and so you know?
Yeah, that's a possibility. So you can, I mean, if the data are the big thing about geometry is knowing if it's locally flat or how flat it is. So if your data are quite concentrated, then any Taylor expansion will be good. But if you are really far away, then maybe you should do something more efficient than just trying to have high order Taylor expansion to be able to reach things. So it's really a question of finding what's the scale or what's the curvature that you can feel at the scale where you're computing things.
Did you bring samples of the Berango for the audience?
No. These are typical candies from north of France.
He did bring some croissant, which all right, thanks, Xavier. All right, so last talk of the session. I'm the last one standing between you and the lunch, so I'll keep aware of that. I'll keep it short.
no you.
So I want to talk about some sort of opportunities we have given the data availability and so on. The focus is on shapes and the focus is on complex structures. Rob Cass is not here, but I included this subtitle like Kendall meets Fisher Robert. So I think that was just for him, but he's not here. So opportunity lost. So I want to talk a little bit more generally, not just neuroimaging data, but imaging data in general. I mean, images are everywhere in every walk of life. So as we are learning and training our students to handle neuroimaging data, why not train them a little bit more broadly? Talk about images everywhere. So the point is, when we look at images, we are not that much interested in pixel values in the image. We are interested in what is represented in the image. What is the object that we are looking at? That's the first bullet. The focus is on objects captured rather than the pixel values themselves. Of course, there are exceptions. There are a lot of people here doing fMRI, and there the actual pixel values have meanings. So one has to be and these are the exceptions in general. In computer vision, pre-processing, there was some discussion about pre-processing. In that context, pre-processing implies extracting objects from images. And that's the main task, segmentation, you can call it, or something else. In that context, any transformation of the image that helps us improve this extraction is a good thing as long as we don't destroy the object that is being studied. Okay. So it's the objects that becomes the focus of our study. And of course, it's a well-known thing, and things that I learned from people like Mahadevan, who talked earlier. Form dictates functionality. That's like an undergraduate class in biology. The shape of a biological object dictates and constrains what that object does in a larger system. So shapes are important, objects are important, and their forms are important. And the last point I want to make is that this object data is not Euclidean. You can't take objects and say, let's add them up. Let's subtract one from the other, and let's average things. So doing statistics is fairly non-standard in this object space. So we have to think fresh. Now, getting on to the shape analysis, shapes of these objects, there is, of course, like people like Laurent and Xavier and Maha, there is a shape community, and people have developed a lot of tools for analyzing shapes. There is a tremendous progress. We go to the shape conferences, you see there's lots of new ideas about how to even take complex objects and be able to understand and quantify the shapes or the variance in shape within a population. So we have tools for things like how to mathematically represent complex shapes. Xavier talked a little bit about metrics for comparing things, for deforming objects. So I mean, this is a complex. So this is a geodesic between one tree into another tree. It's a fairly complex objects, but we know how to do this. We have metrics and we have computational tools to compare, deform one shape into another and compare and quantify these differences. With such metrics, we can even talk about some basic statistics, things like averaging. So as I said earlier, these are non-Euclidean objects. You can't just willy-nilly do standard statistics, but now we have tools to be able to talk about computing means, computing covariances, maybe doing PCA, and even putting some kind of simple models like Gaussian type models. Not exactly Gaussian, but some variation of Gaussian models. So there is a tremendous progress that can be brought to the data sets that we have been seeing in this workshop. One has to sort of connect with the community and understand and keep up with the progress. There are lots of different kinds of shapes. The tools that I mentioned in the last slide, I think this sort of work started with simple shapes, like shapes of simple curves. But now one can talk about, we can do these things. We can compare shapes of 3D objects. We can compare shapes of these trees. We can compare shapes of graphs, networks. I mentioned, I saw it being mentioned in the workshop a few times, networks, complex networks of either neurons or blood vessels. We have tools to now, may not be the best tools, these tools are still evolving, but we have tools to be able to compare and quantify shapes of these complex objects that you're seeing at the bottom. What is needed now? I'm trying to get into the questions asked by the organizers. What we need is more advanced statistical tools, regression models. We need to be able to take these shapes and involve them in regression models in some ways. We know how to do regression models with classical Euclidean data, high dimensional, low dimensional. But how to write regression models with structures of hippocampus or arterial networks or neuronal networks, how to insert them as a predictor to get a response that we're interested in. Modeling populations, more complex models, not just simple Gaussian models. Longitudinal data, of course, we saw this brilliant example from Mahadevan on how cortical surface evolves. You have data at distant times, how to develop statistical longitudinal models for long times, which can capture these evolutions. So I want to just talk about a few of them, and maybe just the very first one. If I can just get the first point across, that's enough. But there is a longer list of things for us to do. But given the time, I think I'll just at least try the first one, regression models, shape regression models. So a lot of people here work with functional data, and you have seen these regression models called scalar on functions, functions on scalars, functions. In the same theme, in the same spirit, we have scalar on shapes, shapes on scalars, shapes on shape. So the shapes of these kinds, either anatomical shapes or neuroimaging shapes, or some general shapes, our goal is to include these shapes in some kind of regression models. So I want to focus on this scalar on shape. In my mind, that was the first natural thing I wanted to do. Turned out that was not the easiest thing to start with, but anyway, that's where we are. So we want to be able to write this regression model of this kind. Yi is a scalar. Inside this, on the right side, is the shape of an anatomical object. So some function which maps a shape space to a scalar, and then perhaps noise. Okay, this is just trying to extrapolate, take any regression model that you have seen. But the problem is that the object that is inserted here, it's the shape of the object, not an object itself. You may represent an object with a function or a vector or a matrix or a tensor. But shape in general is invariant to certain things. You have an object, if you rotate it, its shape doesn't change. It's invariant to rotational transformation. So this part that you're inserting in this should not change with any of the shape preserving transformation. So we have to make statistical models invariant. How are these things done in the past? As we've seen, this already has been a discussion here. The way things are done currently is you take these objects and you register them, you pre-register them in some ways. And once you have pre-registered, you have gotten rid of these transformations, hopefully, and then you apply the statistical model. Okay? We want to do things differently. In fact, I'm going to skip this slide. Looking for a parametric model. So this is, I just mentioned this, just a quick repeat of this. The idea is you have some functional predictors, FIs, scalar responses, YIs. What we do currently is we pre-register this data using some off-the-shelf registration technique. And then we write some kind of a model. Here is a simple functional linear regression, or you can even put some sort of an index function, nonlinear index function H on it. That's what we do currently. In fact, that's what we did also in the previous work. We use these anatomical shapes, subcortical structures to diagnose certain disease and to write regression models. We pre-registered everything. So problem solved. Well, not quite. What we want to do is actually do this registration inside regression. We don't want to treat a lot of this pre-processing with some software under some metric and then apply some model with some metric and some other assumptions. It has to be done unified way. It has to be done jointly. So here is a simple idea that instead of this pre-registered model, what we are taking here is this inner product is now replaced by another inner product where this supremum is removing these shape-preserving transformations. So for example, rotation, if you have to rotationally align the objects, instead of pre-aligning the objects to themselves previously, we are aligning them to this regression function beta, and these are the individual objects. There are some details I won't be able to explain because to be able to pull this off, you need invariant products. Xavier emphasized that. You need invariant products so that these optimizations are even well-defined. And so one of the examples here is not just rotation, but registration. If you have images or shapes that you are trying to use as predictors for a certain disease, instead of pre-registering the images, you register it inside this regression model in this way. There is some nice interpretation. I don't know if I should go that much into it, but for those people who have done some shape analysis, the regression coefficient here, beta, has a nice interpretation to it. In shape analysis, we talk about mean of a shape. You give me a bunch of shapes, I calculate a mean shape by registering individual shapes to that mean shape and then doing some kind of averaging. And that's defined by this first equation. That's the mean of a shape. In the same way, this beta, that regression parameter, turns out to be some kind of an optimization, similar optimization, where this supremum, this registration problem, was inserted in this manner here, and it also shows up in this manner here. So instead of pre-registering and having a mean shape beforehand, this beta turns out to be some kind of a regression mean. It's a mean of those shapes, which is helping you best predict the response yi that we are interested in. Okay, so it's a new way of defining the mean of a shape, not just independently among the objects, but in the context of predicting a certain response. Here is another example. You have a number of functions, scalar functions, and we simply want to align them, register them by themselves. So we have done this. We have done this for 10, 15 years now. We have this time warping, and it aligns these functions. But now, if you want to use these functions to predict some responses, then the alignment of these functions to best predict these responses turns out to be a different alignment than pre-registration. I think this slide is very important, if I can get this across. This is an alignment or registration of functions without taking yi's or responses into account. This is the alignment of functions. This is the alignment of functions with respect to the response variable, taking that information into account. And it's a different solution. So we have done that. We have done it in a number of cases, running out of time, and you get superior results because you are using this optimization. You're using this optimization as a tool to reduce the prediction error as much as you can, as opposed to doing pre-registration. So you in most of the cases, you gain in performance because it's an extra degree of freedom that you are using. There is something I wanted to talk about shape populations. I'm going to skip that. I want to talk a little bit about longitudinal data. I think there was a talk or two earlier about how to model shapes evolving over time. You can take Mahadevan's example about cortical surfaces changing over time and how to model it. This is a particular example from biological growth. These are these tomato plants. Each row represents observations of these plants growing over time. So we have pictures, we have 3D pictures of these plants growing over time, and we want to be able to write down models on that. Again, I want to explain the problem because I don't quite have a solution. One way of doing this, of course, we want to treat it as a stochastic process, model it, and so on and so forth. One is to take this process on the shape manifold and in some ways flatten it, make it into some kind of a vector space and apply stochastic modeling on the vector space. That's one way, that's the right approach. On the left, we do it intrinsically on the manifold, write stochastic processes, time series models, or some functional data analysis type stochastic models. We have done the thing that I mentioned on the right, somehow flatten the space and then write something in functional data analysis. So with Zhenghu and Hongtu, this was the work on modeling the growth of subcortical structures. But this belonged to this right category. Somehow flatten it, simplify it, use existing tools. But more natively, more accurately, I think it has to be done on the actual spaces and respect the geometry and exploit. Okay, I think I'm running out of time. I'm going to just stop here and thank you very much for your attention. It says -26. If you have a question, you can ask me, or if you have a question for any of the other speakers, this is a good time to ask question or two. Hernando.
Thank you to all the speakers. Really very fascinating, very informative talks. So Anuj, I have a question for you. Again, very nice talk. When you had your regression model, okay, first you have everything pre-registered, and you said that's not quite the best way. So you had the other approach where you did the optimization. Yes, this one. Yes. Yeah. So at first I thought it was a bit unnatural. I thought you were going to put it on the, you were going to find these transformations gamma I by minimizing some error. Can you elaborate on that if you have?
Yeah, so,
if you get my drift.
right, yeah, exactly, good point. So the way these registrations are done, we are registering individual objects to the regression function beta, and beta is chosen by minimizing the prediction error, the overall error. So it's the beta that is trying to minimize the predicted quantity and compare it with the response. But the registration is inside this. It does contribute to the overall problem, but the immediate way it is done, it is you are aligning individual object to this regression function. So this sort of replaces the classical inner product with a new inner product. So it's a swap. This inner product with this inner product, which contains the registration.
I thought that could you have put the optimization of each of these gamma i's outside as well?
I think one can do it, but I think ultimately it's a question of what metric goes here. And if this metric remains same, the gamma will come inside because gamma will not affect it. One can change this. Onto.
Your purpose is
Ask somebody else. Good.
if your purpose is doing regression, maybe I don't think you feel that the retration, the strict retration, the exact retration is necessary. Because you're changing the problem into minimax problems, it could be very unstable in practice. If your purpose is just doing predictions, you can, release the space a little bit and make it the regressions more better.
Okay, I won't answer that, but I'll just say something else, which is why we do registration, because the variation across populations in neuroimaging data often is subtle. And it's less than even the registration errors we have. Registration errors, if you don't do a good job of registration, it dominates the variability across population and you lose the power of your test. We want to do registration for that reason.
I know, but sometimes the registration you introduce the bias because the data is
Right. So that's why in the regression context, you want to do regression with that point of view.
Yeah, that's the reason I'm saying that if the purpose is predictions, and actually you essentially if you relax that operation, you maybe open a lot of space to do better for prediction purposes.
Maybe what you are saying is that, I mean, it depends ultimately what is your loss function for the overall problem. If my loss function is prediction error, then I want to use the registration to minimize the prediction error, as opposed to not doing it, because I have an extra degree of freedom here.
Yeah, we can discover it.
Or maybe you should go Bayesian and look at what is the error that you're doing at different registration, perhaps weigh differently the different possible registration.
Yeah, that I completely agree. I think that's a structure one can impose as opposed to sort of optimizing, one can integrate it out with the prior. I think Marina talked about that too. So I think that there's a thought process there, one can do it. Right. John.
Just if I'm understanding, so is there a risk of overfitting here with the registration? Do you need to cross-validate around it in any way?
I think that's true with either way, either you do it beforehand or after. I think that same consideration has to go into effect about overfitting and validation. This is just simply two different ways of doing it, and that's
The
where.
second one is matching to your outcome,
Yes.
whereas the first one, you're
Well,
independently doing.
Yeah, you independently register. You take somebody's method, download somebody's image registration toolbox and register images.
It makes also cross-validation easier. What scares me with your approach is that if you want to validate Bootstrap or anything, if you end up with a very heavy proposition.
Heavy, yes. Yeah.
Anuj?
Bootstrap, because there is an extra optimal. It is computationally expensive, for sure. I didn't say a word about defending that. Right.
Can I ask a question?
Oh, yes. Go ahead.
So this is a question for both Xavier and Anuj. So I like very much the perspective that you bring by emphasizing the need for invariant measures for shape. And of course, that's exactly what, if I was to approach it as we do, from a biophysical perspective, we also want to do. If I want to create a generative model, if I want to write down either dynamical equations or minimize an energy, we have to respect exactly the same kinds of invariances that you're already talking about. Invariances associated with maybe the Euclidean group, maybe the similarity group, maybe some other groups. And so a very natural question is, do you think that this effectively is going to lead to a convergence where the physical or the biophysical approaches which build in the invariances in the context of generative models and the statistical models which you build in the invariances into your metrics will converge to the same endpoints?
I'm going to give this to Xavier.
Well, tough questions. It's I have no clue. I think if there is a good model, I mean, if we take the statistical hacks, there's no true model. So, but from the biophysical point of view, we believe that there are true models. So maybe at some point the models that are useful could be the ones that or could converge to the ones that are biophysical.
So let me say that even the biophysical models, there is no real truth. It's a question of scale dependent truth. You know, if I look at morphological morphological evolution of shape, you know, I wrote down a very simple free energy which was associated with a quadratic form in the deformation gradient. Now, Anuj knows, and perhaps Xavier, I mean, Anuj and I have talked about this in the past, but this is a question for the community is, you also, when you want to try and build metrics for comparing shapes, want to preserve similar kinds of invariances. And so if I write down these minimal distance forms or metrics in terms of appropriately and properly accounting for invariances, don't do L2, do Fisher-Rao or something of that sort, I have this vague sense that we will converge. How can it be different? If you're building the invariances in to understand real data, which has structure, and I'm looking at it from the point of view of creating a generative model, which leads to a prediction, why would you expect there to be a real difference? Or would you? Maybe I shouldn't pose the answer, I should ask.
Well, just to add to this, just to continue the questioning/answering you're doing, if you look at the classical metrics for something as simple as functional data analysis, I shouldn't say as simple, it's a very exciting topic in statistics, functional data analysis.
No, you can say as simple.
The classical metrics are degenerate. They are degenerate. You cannot use L2 norm to do registration.
Yeah, but if you think about what you and Eric and, going back to David Mumford and all these people and Ulf Grenander, when you start thinking about Elastica and Elastica-like, it's not a surprise that those models appear when you start thinking about invariances, just as it was not a surprise when Euler wrote down the simplest functionals based on the square of the curvature. I mean, it was not, you can say it's physics, but it's essentially a mathematical abstraction of some very simple invariances. I need invariances associated with translation, rotation, but not scale. And the first thing that appears would be the curvature. So am I overstating or oversimplifying?
You never do. No, I think, I mean, at least it's a very biased opinion, and, you know, we have a whole community has pushed forward these elastic metrics, and we are trying to bring it to the statistics community, that the metrics and the tools that have been used traditionally for so many decades in statistics somehow start failing the moment you start talking about simultaneously doing the registration.
Because you have
It's-
very structured information.
Yeah, and you want to exploit that. I think you want to respect. I think that's the very minimum. I thought your question was among the family of such metrics, like Xavier showed so many invariant metrics.
Yeah.
Among those, which one are relevant?
That as well, but that would
Right.
now become phenomena specific. So it
Right.
depends on which, at what scale and where in biology you're looking at. I mean,
Yeah.
you know, I don't think there are universal models in biophysics either. I don't think that, I mean, I would love for there to be a well-agreed truth, but there isn't. It's scale dependent, depending on what you're looking at.
But it's a good question. I think, I mean, that's part of the message we had for a long time. Jengu, you had a question?
Yes, I think this is a question for you and Xavier. So from computer science community, they have developed a lot of generative models, which can work really well to generate images, videos, and shape data. It seems that those models has the power to disentangle the geometry in those complex data. So what's your view on those models and how they're going to help us to understand, the space? Do they have power to, flag the complex Riemannian manifold? And then we can do statistics in, Euclidean space and then use decoders trained by those models to, map back to the original manifold space, etc. Compare with current complex Riemannian mathematics we develop. What's your comments on this?
Well, it's a little bit the question of how much data do you have. So for me, if you're in the big data domain, then it means that locally, I mean, even if you have data on manifold, if you have an infinite number of data, you're going to have enough data close to the sample where you are, so that you don't need to know anything about curvature or whatever. It can be locally Euclidean. Now, what's happening, and that's what I've seen in medical imaging, is that when we arrive in this setting, then we have lots of tools that works, and this is not research anymore. What's going to be interesting is maybe the subtype of the disease that make a difference. And here you're going again from, I don't know, tens of thousands of data, which is never happening in medical imaging, to 10, because you want to distinguish things that are very subtle. And here, again, when you're going to the low sample size, here the geometry may make a difference. I say may because that's just models. But if you model correctly, what are the assumptions? Then, I mean, if you want to interpolate between the path between Paris and New York, it's not going through the center of the Earth. It's basically, if you know a little bit, then you're going to go to North Pole or close to that. And it's just a generic global information. And that's not something you can guess if you don't have the data. So this is really the type of things I believe that invariance is giving. It's a way to compare things which are far away. I mean, if it's close by and you've got lots of data, there's no need for geometry.
Sure. Yeah. I think I can just add to that, so far, basic neural networks, Euclidean loss functions, standard, this been done. But now people are bringing geometry into the loss function in neural networks. So one is going to gain something by bringing in invariant metrics or structures inside the network. I think it will improve, just like we improved the standard analysis.
I think the geometry does help with neural network to design the architecture. Is in terms of the complexity of the functional class, because you can look dramatically lower down the MSE for that, and the theoretical bound is much better. And also if you have the geometric constraint built into architectures, actually the performance is much better, because when you incorporate geometry, actually the training data set you need to
is much lower. And because you incorporate the geometries, you don't need to search the whole space in order to cover that.
You don't need all possible rotations of an object if you have rotation invariance. So
I have another follow-up comment because I think that for SPD matrices, you develop the log metric and also there's a geometry, these both are kind of practically log metrics have been widely used because computation is simpler. That's the reason it become popular. Because to me, I think the most important thing is computational, that's the most important consideration compared with other theoretical beauty, right?
in the near future.
No, I think in practice, that's what we have been using a log metric for. To me, because you just need a basic measure, then you can quantify anything, right? Then you have a distribution on that. To me, I know the people argue about basically you have different metric how to choose it. There's no criteria for that, right?
Yeah, it is a metric versus model question. I remember when David Mumford first talked about using different metrics for curves, he used to say we don't have to be very precise about the metric because we still have a distribution to impose which we control and we can take care of some things there. But it is clear that the mathematics has to be at least in the right ballpark.
No, I'm talking about the basic, you choose a good ones.
Yeah.
Computation is simple.
Yes.
And everything else basically using distribution to pull
Let the distribution.
right?
How about we let them, you talk and we go to work?
I'm hungry too,
But maybe just a point, which is that having efficient methods is nice, but understanding how you can use them is also important. If you take a linearization on the sphere on top or on the North Pole or South Pole, you're going to get very different results. But if you understand that there is a global thing and maybe you should recenter, then maybe you can do things much more easy. Have the best of both worlds.
All right, thank you very much, everybody, and thanks to all the team.
Lunch will be downstairs and please, next section will be 12:45. Thank you.
Thanks, Anuj.
See you at lunch, Ma.
Bye, Laurent.